Dear colleagues, dear organizer, I'm honored to lecture again at such a prestigious congress here in Belgrade. In today's lecture, I will present to you a study of our research group, Enhanced Hemodynamic and Clinical Response to Alpha-CGRP in Migraine Patients Measured by TCD. It is well known that CGRP induces vasodilatation and enhances neurological transmission. Therefore, the level of brain sensitization could be reflected in CGRP-induced brain hemodynamic changes detected by a transcranial Doppler. The vascular biomarker CGRP could be a useful clinical tool for assess sensitization interictally and to determine treatment response for monoclonal antibodies. Until now, only a few studies have investigated exogenous alpha-CGRP response of brain circulation. Some authors found decreased mean arterial velocity in MCA during alpha-CGRP infusion, indicating a dilatation of MCA. The same group found the increase in plasma CGRP cause headache in patients with migraine. Available studies did not compare the response to alpha-CGRP between healthy subjects and patients with migraine. In our study, we expected an increase of MCA vasodilatation to alpha-CGRP in migraine along with the occurrence of the CGRP-induced headache. In the study, we included 20 healthy subjects which represented the control group, and 20 patients with migraine represented the study group. We did not find statistically significant difference in sex and age between the groups. TCD with 2 MHz ultrasound probes was applied to measure mean flow velocity in left MCA through the left temporal acoustic window. During the entire experiment, mean blood flow and heart rate were measured using non-invasive plethysmography. The end-tidal CO2 was measured by a ventilation mask and infrared capnograph, which was connected to a computer. End-tidal CO2 signals were recorded on the same time scale as other variables. TCD Multidop X4 software was used to define mean volume of VM MCA, mean arterial pressure, heart rate, and end-tidal CO2 during 5-minute intervals. One interval during the baseline period, 5 to 10 minutes point. This was point 1. Then 2 intervals during the alpha-CGRP infusion, 15 to 20 minutes. This is point 2, and 25 to 30 minutes, point 3 of the experiment. And 1 interval after the end of alpha-CGRP infusion, 35 to 40 minutes of the experiment. This was point 4. The mean VM MCA was calculated for each 5-minute interval. The mean values of the variables mean arterial pressure, heart rate, and end-tidal CO2 were also calculated for the same time intervals as VM MCA using TCD software. Determined responses to alpha-CGRP as the differences between measuring points for VM MCA, end-tidal CO2, mean arterial pressure, and heart rate. The response 1 represented the difference between point 1 and 2. The response 2 between points 1 and 3, and the response 3 between measurement points 1 and 4. The incidence of CGRP-induced headache was recorded within 1 hour of the experiment, This was immediate CGRP-induced headache, and within the next 12 hours after the experiment, this was delayed CGRP-induced headache. The number of subjects enrolled in the study was driven from previous hour studies. For statistical analysis, SPSS version 21 was used. First, we analyzed the occurrence of immediate and delayed CGRP-induced headache in healthy and migraine patients. An immediate headache was found in 7 migraine patients, 35%, and in 3 healthy participants, 15%, with a statistically significant difference between the groups. While delayed headache appeared in 13 migraine patients, this was 65%, and while only 1 healthy person, 5%. Migraine was significantly related to immediate CGRP-induced headache and delayed CGRP-induced headache as well. Secondly, we compared signals of parameters obtained from the migraine patient and compared them to the control group. Figure 1 presents reconstructed signals for VMMC, this is figure 1A, and tidal CO2, figure 1B, mean arterial pressure, figure 1C, and heart rate, figure 1D. I now showed significant differences in VM-MCA response 2, but not in MCA response 1 and MCA response 3. The response of end-tidal CO2 was significant for response 1 and response 2, but borderline for response 3. Mean arterial pressure showed significant differences in response 1 and response 3 and borderline in response 2. Heart rate between the groups was not significantly different by any response. The relationship between VM-MCA and tidal CO2, mean arterial pressure, heart rate, and migraine was tested using logistic regression. We found significant association between migraine and VM-MCA and tidal CO2, mean arterial pressure, but not for heart rate. Finally, we tested the relationship between CGRP-induced headache and physiological variables using logistic regression. The CGRP-induced headache represented the dependent and VM-MCA presented the independent variable. Logistic regression showed the association between immediate CGRP-induced headache and VM-MCA response, but not between delayed headache and VM-MCA response. Interestingly, we found a statistically significant relationship between end-tidal CO2 response and delayed CGRP-induced headache, but not immediate CGRP-induced headache. In conclusion, alpha-CGRP could induce vasodilatation of MCA probably via activation of trigeminal vascular reflex. Decrease of end-tidal CO2 might reflect a compensatory decrease in arterial partial pressure of CO2, which underlies the normalization of CBS during the CGRP activity. It seems that immediate CGRP-induced headache are caused by the direct CGRP effects on neurovascular structures, while delayed CGRP-induced headaches are an indirect CGRP phenomenon due to compensatory processes. CGRP had chronotropic effects on both the migraine patients and the control group, while systemic hemodynamic was not importantly affected in either group. At the end, I'm inviting you to two virtual meetings in Ljubljana, one in October and the second in December. And thank you for your attention.